presentation I will do today, it's something that comes from a meeting I have with people from Fisheries in Ocean Canada, from also Parks Canada last August, and I realized that there was a lot of misconception of the Arctic of pure element. And I didn't know how to raise that and to build my case to say, look, and you will see in a few minutes why. And I'm sure if I'm asking, do you think there is less biodiversity in the Arctic than in the Gulf of St. Lawrence, for example? And I'm sure everyone will, just for fun, do you think there's more species in the Arctic or in the Gulf of St. Lawrence, which is more warmer? Most of the people will say, no, there's way more in the Gulf of St. Lawrence. And that's based on a few assumptions, which it seems true, and I was the first one to believe that until I started going to the Arctic. So one of my goals during this meeting, that we were talking about creating new marine protected area in the Arctic, I just realized that a lot of decision makers got some misconception of some aspect of the Arctic. So I decided to bring some paper all together, some of my labs and some other labs. So I'm stealing some really nice work from Soldier team to try to make a case about 4 misconceptions that I will try to present to you. And using this conference again to try to build the case in a more stronger. So all your comment at the end, question are more than welcome. That's the goal would be to create some clear, I would say, comment to bring back to people in Ocean Canada for any decision maker, at least for creating marine protected area in the Arctic. Having said that, and I need to apologize right at the beginning, I'm colorblind, so the pointer is red. My worst color is red on blue, so I may miss. So sometimes I may point the wrong place. Sorry, straight at the beginning. So one of the major misconceptions that I realized was a lot of people say very low marine biodiversity in the Arctic. That's what we have in mind straight ahead. However, during the Census of Marine Life, maybe someone knows this large program from 2000 to 2010. So we have a program in Canada, and we decided to just compile all the biodiversity information we will find at that time. So when I just make a summary of the table here. So we look at, I just put a few major taxon or taxonomic group, and I split that in 3 regions. Maybe I should just move the. Perfect, thank you. And as you can see, when you look at some major group for that we have in mind marine mammal, there's typically less in the Arctic compared to the Atlantic Canada and the Pacific Canada. When you look at phytoplankton, suddenly, and we did not include the Hudson Bay, that should be enough. You have close to 1,000 species only of phytoplankton compared to 600.56 in the Atlantic, same thing with the Pacific. And because of a very ecologist, I'm looking at the animal living on the sea. So when we look at something, specifically the animal living inside of the sediment. So when you look for 53 meters squared at that time, we managed to collect 53 meters squared. It's probably, that's surprising, but that's probably this side of the room on the bottom here. That's not that much for the Canadian Arctic, but we were more than 1,000 species. When you look at the Atlantic during that program, we have way more, 178 meters square, and you can see that's only 100 species more. And when you look at the Pacific one, it's 800, but we have only 20 meters. That was not enough to really make a nice comparison. So we can believe that even with this low number, so there's still a lot of species in the Arctic. Same thing in the Atlantic, but not that high for this size, and 800, probably way more also in that kind of Pacific. So when you look at all the compilation, so when there's another taxonomic group, they are not on the list here, but we were at more than 2,800 species for the Arctic, 3,000, though it's only 200 different at that time, but way more knowing the Atlantic, it's way more known than the rest of the 3 oceans surrounding Canada. So at the same time, we built on the Pan-Arctic vision of the Arctic. I'm not going to go into detail, but during the time when we're doing the Canada, when we're doing the Pan-Arctic lab, so we managed to bring 7 different countries all together to compile their data. And what you see here is the different areas of the Arctic. The number here are the number of samples we managed to compile. And as you can see, some areas such as the Barents Sea is well covered when you look the internal part of Canada, for example, here, this area, we have very few samples at that time. And same thing with Eastern Greenland, Labrador, and you can see at that time when we compiled these, there was not that many except for this area, which correspond to, I'll try to just without doing too much. Sorry, sorry everyone on the, I just want to be sure that we're going to. Should be okay now. I apologize. Is it better? You're not going to see the title. So it's only the number of sources, the number of stations we managed to compile. And from the time 1956 up to 2009, so the study started in 2010. And rapidly, I'm sure everyone of you are familiar with these rarefaction curve of species or taxon, but we have all the different area of the Antarctic, the region, and as you can see, the curve, there's no plateau yet, there's no asymptote level. So even if we sample more than 1,000 samples in some area, it's still going up, and it's only for 3 taxonomic groups: the mollusk, the arthropod, and the sea star or echinoderms. So at that time, to be sure that all the sample, every region got the same makeup. That was the goal. Otherwise, some of the group will be not similar. So definitely, there's still the curve is rising. So, and with the estimation time using the Kinos index, we're close to a little bit more than 4,500 species. So just in small detail here, so we said definitely there's a bias through the region which has the highest proportion of sample. So when we look, which is the vibrancy just here, and you can see there's close to 600 species at that time, or expected species were close to 600 in this, and the lowest one is Western Greenland, which fit with the number of samples. There was only 19 samples, which typically it's not rocket science, only that we have only 19 samples, so we have less species diversity, which is pretty easy. But at least that shows us a general picture. And surprisingly, that even in Canada, the Amazon Gulf, the Université, you can see they were quite high with still very few species, or few samples, sorry, with only 20 meter or 53 meter square of the steep one. That's during that time, so because I'm doing also sometimes some taxonomies, which I love to spend time in front of the microscope. So we discovered plenty of new species, as you can see here, and we're publishing these new species that were seen by scientists or in general. For example, this one, I love this one because it's a polychaete worm. We give it a Inuit name for this tool, which just for your general background, it's only to make holes inside of bone. So we're using that as the sort of plane to make it turn. So, and one of the criteria is exactly the same shape here. So we decided to give this name. So it's only, and we have for the first time discovered some acorn worm, which never seen in any Arctic or polar region. There was 2 new species never seen before. Some of these, as you can see, this one can reach up to 30 centimeters. So having said that, so we discovered so far every summer new species never seen before. And for a long time, the people said there's no endemic species in the Canadian Arctic, which is not true. This is what we're finding each time we're going there. Just for people who don't know that, the Canadian Arctic represents 10% of the coastline of the world. So when you think about it, that's really a large area still undiscovered by many scientists. So more recently in 2020, we decided to be more specific following the study that I mentioned before. We decided to use and try to be the more similar with every sample that we collect, try not to be too large. Look again at the 3 oceans surrounding Canada. And this time, so we look at the Arctic, again, Pacific and Atlantic, for only the Infauna species living inside of the sediment. And this time we used exactly the same method that was during the Canadian Healthy Ocean Network program. So we were more similar in effort in every region. And as you can see this time, so we changed totally the relationship I showed you earlier. And this time it's the Pacific. When you look at the number of species, the Pacific is way higher. compared to the Atlantic and the Pacific. The Arctic is just in the middle with more than 1,000 species, only of species living inside of the sediment. So definitely we switch because we have now a more similar effort of sampling. And that's where I try to destroy what I mentioned about the misconception. So when you look at the Shannon Diversity Index, suddenly it's the Arctic, which is way higher than the Pacific and the Arctic, only for infaunal species living in this inside of the sediment. So that was traditional taxonomy. So at the same time, in collaboration with the laboratory of Louis at Laval University, we decided to do and try to work with eDNA for invasive species, but at the same time doing a full array of the different species. So we went to different areas, 3 ports in the north, in the Canadian Arctic, Churchill, Deception Bay, and Iqaluit. At each station, we sampled the eDNA from the water. We look at also traditional taxonomy of zooplankton and same thing with benthos, exactly at the same area. So basically, I was diving, collecting the water at 1 centimeter, 2 centimeters above the seafloor, and I was taking a graph. So the goal was to see if we have a connection among these, basically to try to avoid traditional taxonomy, who may sometimes take more time than eDNA. That was the goal behind that. So what you have here in the graph is the traditional sampling, the eDNA, and the common genus. And as you can see, surprisingly, when you look at the different regions, we don't have so many common species among the eDNA and the traditional taxonomy. So we were really surprised with that. We were expecting, and I remember then sampling above a crab, and we have no signal of crab in eDNA. I was sampling 1 centimeter above. So we have it in our graph, but not in the eDNA signature. So it was really surprising. And when you look at both, so the eDNA got for Deception Bay quite a high biodiversity. For the eDNA it's here, the traditional taxonomy, it's always higher than the eDNA. So that was really a large surprise in our case. We're expecting totally the opposite. eDNA will sample way more broader than the traditional way. What we realized then, we have some groups which are really more present in the traditional way compared to the eDNA. And we have the subsample here of some of them. For example, Bryozoan, Brachiopoda, and Foraminifera were more present in the traditional sampling compared to Cnidaria, Porifera, and these groups here in the eDNA, showing a complementarity instead of one replacing the other. So we managed to increase the number of species in these 3 ports. And as you can see, we were having only 87 species which were common to the both method. So, and more than 300 and this number here for the 2. So really it's this gamma diversity, we're expecting to have a more broad view, but basically the 2 methods have a narrow view when we had both, way better view for the biodiversity in general. So, and in this case, for this group, we have more than 500 genera of Arctic marine invertebrate only for that sample in 3 ports, which is going to complement what I mentioned earlier. So that was for my first argument about the misconception of low biodiversity. I hope I convinced you a little bit. Then when you compare the Arctic, yes, definitely when you look at marine mammals, when you look at fishes or macroalgae, there's more in the Atlantic and the Pacific. But as soon as you enter the phytoplankton, the more smaller organism, it's a more subtle view, and some groups are even higher in the Arctic for phytoplankton, for example, because you have the ice algae and the phytoplankton. This is what our studies seems to show. The second one I heard, the second conception, it's usually when we're going to remove the ice from the Arctic, we're going to move from a system that was beneficial for the benthos, the group of organisms I'm studying, to a more pelagic-driven system. A lot of people in this meeting were having in view, for example, the Gulf of St. Lawrence, where I have only 4 months of ice, 3 months of ice during the winter, and the rest of the year it's free of ice. And you have a system which is more pelagic-driven compared to benthic-driven. That's the term they were using. So, but however, you will see. So that's something you probably have seen before. That's data from yesterday, from the National Snow and Ice that have entered. What you have here for the people who are not familiar with these graphs, That's the North Pole, the Arctic. You can see the orange line this year. So it's the median ice edge from 1981 to 2010. And you can see some area here and here. So the ice extent is getting smaller and smaller all the time. And you have here the same type of graph from August to December, where you have the minimum ice extent, which was in 2012, and you have 2022 just here in blue. And we almost reached the same level, the lowest level of 2012 here in mid-October, and it's getting back to a normal inside of the. So showing there's less and less ice in the Arctic through here. So for people who are less familiar, when we say that it's going to start from a benthic driven to a pelagic driven system, I will take a few minutes just to explain. So we have here, it's the old season where you have the ice cover, the ice algae, and at some point when the ice breaks, and so you have a bloom of phytoplankton, but at the same time, all the ice algae under the ice are fluctuating and falling faster on the seafloor, so bringing some new material to prevent us. And it goes like this. But if you have a longer pre-season, you have this or ice algae bloom at the beginning, and the rest of the season it's extending, and you have less large bloom, but the bloom is longer, and the zooplankton will benefit from that. So here it's a lot of ice algae, less phytoplankton. The benthos will receive all this material more rapidly, and here on this graph, with less ice time before phytoplankton, the zooplankton will benefit from that. So that's what is behind, and that's why I was in this meeting. The people were saying that we're in the Arctic moving to this system more and more. There's also one study from Soren Risgaard and Ronnie Glue in 2007, which in their case, they said that's exactly something similar. The ice period will get longer here compared to here. You see that again in this system, the benthos will benefit, but in their case, they show that there's a mismatch with the zooplankton because the water is still cold enough and the zooplankton is not emerging faster. So even though you have a longer period, so the phytoplankton is all reaching the seafloor also. But this has been done inside of fjords of Greenland. So the goal was to test one of these two hypotheses. So to test this hypothesis, we decided to use bioarchive. And this little bivalve, which you don't see here, but it's a bivalve who can live more than 150 years old. So even if it's only a few centimeters, and we're using exactly the same thing as the growth band inside of trees, but you're using the same thing, but on a shelf like this, and we can measure and calculate their growth through time. So we decide why we use that, because it's up, not moving. Wide distribution at the full Arctic scale. It's a filter feeder, getting what is coming from the surface. So that means we can separate what is from the seafloor from the phytoplankton if the phytoplankton reach the seafloor. And at the same time, it's a long-lived species. So we collect them in few areas, but one specific area we're going to study with a box score. So this is the different area where we found them in the Canadian Arctic. And more specifically now, we're going to look at sclerochronology, which is the pattern of growth of the animal. And what we call sclerochemistry is the study of chemical elements inside of this different layer of age through time. And we sampled vividly in the now Polenia at a depth of close to 600 meters, 568 exactly, on October. So that's where we collect plenty of little animals. And the Polenia is in the right area where we know there's some heights, there's some movement, and it's inside of a future marine park. So I just want to compare with this really nice study from Simon Bélanger, Marcel Levin, and Jean-Éric Tremblay, where they look at exactly, so the star here, this is where our animals are coming from. And this is they use seaweed data, 13 years of seaweed data time series. And when you see here in red, so that means there's more production than usual. It's a more early bloom in the area of phytoplankton compared to June. Then usually the bloom is less important now, but it's earlier than before. And when you look here in more in July, there's almost still the bloom is
less important than before. It's what they brought. It's only, and it's starting way earlier because of the ice breakup, way earlier in time. And also at the same time, we were lucky that you have in this area. So a current meter, thanks for the Hamilton Science Group, they are renewing the mooring in this area since close to 15 years now. That means we have some real data also of temperature, salinity, and every parameter along this mooring at 1 kilometer from where we collect the animals. So just rapidly, the species we have, it's up to the oldest one we have in our sample was 103 years old. So that means we can go back in time for their growth for 103 years back in time. We cut the shelf like this, and you have some growth band. That means it's an increment of during the year of the animal, and it stopped during the winter. So we've done that with plenty of animal, 15 total. And what you have here is the standardized growth index. So how to read that from 1950 to 2010. So each time you're above 0, that means the animal is growing better than we expect from the model. And when it's low under, that means it's growing less, it's less beneficial. So as you can see here, it's suddenly since 2000. So the animal, there's some variability which goes with the different oscillation of the Arctic. The Arctic oscillation seems to fit this and stop to fit the model here of the growth of the animal starting in 2000, where the animal seems to have always a better growth, and it's match also the sea ice pattern. So that means that the animal is growing better. So we look at it through sclerochemistry now. Is it only the fact that the water temperature is higher or the salinity? Then we know they can affect how the animal will behave and grow. So you don't see it correctly here, but what you have is the time. Yeah, sorry, just focus on the top one. So we will expect that at the end here, through time, you will have an increase, a difference at least on the oxygen stable isotope ratio, which is it's a good proxy for temperature and salinity. And we look also at the current meter or all the mooring of the Hamilton Science, and there's no clear pattern in the last from 2010 of an increase in temperature. So it seems that it's not totally the temperature, which is really the it's a stable ecosystem there. So probably the temperature is not the key element here. We look also from Thibault et al. In 2009, it's at the barium calcium ratio inside of the shell for every year. So we will expect that if it's the food, and you see suddenly it seems to have a little signal or more variability since the end. So is it the signal? That was a question we had at that time. So potential then from the chlorophyll, why there's a better growth. There's a strong interannual variation, but probably, when you look really at the end, it seems to start to have more. Sorry, I need to point to see where I am. Okay, you have more variability. That may be a signal why there's a better growth also. But it's not perfect, but there's some suggestion going this route. So I'm not the expert in fatty acid. If you ask me any question, I will refer to Christopher in Mon and Memorial. But I like to show this graph, which is not published yet. So it's only to show them when they look at inside of the same type of animal they were there, and it seems to have more signature inside of these fatty acid for really when you look at dinoflagellate and diatom, and very few for the copepod or even for the suspension or the sediment. So definitely, I mentioned I'm not the expert. I'm stealing the data from Christopher. So it's only deeply suggests a contribution in microalgae, mainly diatom here. So compared to the other aspect, still suggesting then from 600 meters, there's no zooplankton eating the phytoplankton. A lot of this material reached the seafloor by 600 meters, which is also what we test now. So we look at the study of Blais, showing the chlorophyll A at different level, exactly where we collect our animal. And you can see there seems to have a decrease they don't have the data. This is where they stop in their paper, but we need to explore this area to see if there's an increase or a change here. And there was this, to be sure that what I mentioned in the zooplankton is not there, we need to look at the zooplankton definitely. So this is what the laboratory of Louis was doing until Louis passed away 2 years ago. So it's still on, I will say, on the ice, if I make a really bad joke. So we're waiting to see who can continue this work, and we're looking to find some funding. But as you can see, they were the sample analyzed at that time, and you see there's no pattern. Then zooplankton is not increasing really for the same period. Then our bivalve on the seafloor increased their growth rate. So still suggesting a mismatch between phytoplankton reaching the seafloor. There's no zooplankton in the middle. So eating the food, it's reaching the seafloor. So that's my idea here of the mismatch between primary production and zooplankton. So what do we expect? So that was my second argument to try to say, no, it's not going to change that much from a benthic-driven system to a pelagic one, because it seems that the benthos, at least in this area, at this region, the benthos still benefit more than the pelagic system, at least for the now Polinia, with what we have so far. So it's still going there. So, but maybe this pattern is only for this area. We need to expand this hypothesis here. So the other one, it's as a benthic ecologist, we always hear that the benthos, it's a low trophic level. So we should focus conservation on higher trophic level. This is what we hear most of the time. And I said, yeah, but in my case, I believe some benthic animal could be at the higher trophic level, while we're saying they're always lower. So that's the usual Arctic food web published by Darnis and in climate change by the CAP, so the conservation of the Arctic fauna and flora of the Arctic Council. So as you can see, I think you've seen this graph maybe if you work in the Arctic, but the benthos, it's a general box, almost a black box, and you have all the link to the higher trophic level. This is what we mentioned, up to human, and how the Inuit and different food provision, and also for their cultural aspect. So it's a paper, and I'm really happy to tell you it has been accepted this week. So I changed this little submitted to Impress. So and in this time we look specifically in the region of the Southampton Island, north of the Hudson Bay. We sample all around the island, and we look in collaboration with the Inuit there, collecting polar bear, ring seal data, narwhal, walruses, every group we can. They are listed here, the vertebrate one and the invertebrate, and we look through different isotope analysis. What are their trophic position? So when you look here from the pelagic, you have the pelagic and benthic, that in this case you have invertebrate, and the benthic, it's really wide. And as you can see, you can go up the food chain with the polar bear really at the top with the ring seal, reaching a level of 5 and 6. But suddenly the benthos, or mostly benthic animal here, so you can see you're from 1, 2, up to again 5 and 6. So sponges and sea star were at the top in this case. So the goal here was to mention that it's not always a lower trophic level for the benthos, and we redrop what we have seen before of the food web with this new one, trying to show that you can have a full benthic food web from here, from the different level from 1 up to 5, 6 with sea star and sponges, with a fueling between the pelagic one and the benthic one through, for example, walruses or eiderduck and a few benthic fishes. But definitely you have two systems, which was never really, I would say, it was demonstrated, but never as clear as this one here. So that was the second aspect, or the third argument about, yes, if you want to start to look only at higher trophic level, you need to consider sponges, sea star, etc., because they are also higher trophic level. So the last one, misconception, it's mostly about the food resources for Inuit community in the north. Most of the people mentioned that they are eating a lot of beluga whale, seals, etc., Arctic char, which is totally true. That's the main food source. But we found this this paper, which is based on the Inuit perception of the organism in the ocean, and that the study of Rapinski, they look at 36 different villages in Nunavik and Nunavut, do some survey, and suddenly realize that a lot of these animals, what they call the Miutai species, closer to the shore, are really, really part of their food, which is mussel, kelp, sea stars, etc. They are eating them. Why? More and more presently, because of the ice safety condition. Usually they are using the ice as almost an highway to get to their food source to hunt, etc. But now the period is less certain, more risk. So they are getting more and more of their traditional food on really on the coastal aspect. So it has this picture here, and it's really eating some kelp, mussel, raw mussel, and even this amazing video from the New York Times a few years ago when they were in the Ungava Bay, where is the largest tide in the world. The people are getting under the ice at low tide with ladder and collect mussel, and they have 2 hours to get out rapidly before the ice and everything start to move again. So they're getting their food sometime under the ice like this. So if you didn't know, that's not the Bay of Fundy, the largest tide in the world. Yes, sorry to destroy your so what we have done, and it's sad that we don't see it at the top here. So yes, they're eating larger than that, but is it going to be beneficial for them? So I'm not leading this program. It's Mélanie Lemire from the health group at Laval University with a PhD student, a master's student, and a chemist Teraya. So they look specifically at one compound called the selenium. The selenium, it's really one of the form of or selenium. It's one form of selenium. Selenium is really good, and that's great. You can see this part. It's the reduction of bioaccumulation of mercury. So that's really helpful for the Inuit because mercury, it's really negative for your health. But when you eat some specific compound, selenium is one of them, it's helping to reduce the effect of methyl mercury. So the Inuit gets 75, the number you don't see, it's 75% higher than any other Canadian inside of the blood of the Inuit. So that means selenium, it's a really high, and selenium also. The maktak, so the skin and the fat of the beluga whale, it's nearly 50% of the selenium for the Inuit come from the maktak of the beluga whale. So here it's only the graph showing, so the team look at the selenium, this compound, and the mercury, and you can see it's all marine mammal. The beluga skin is just like rocket sky. So when you have higher than 0.5, it's really, really high. Or that means it's really good. When it's above 1, it's like exceptional. And where you're just above 0, that's fine. But the more you get, the more it's really nice for the. So just to raise here, then the beluga, it's really high, followed by the walruses, which is really high also, but all the other, it's really, really low. And you can see also the mercury level, which is high also in the muscle and the liver of the beluga. So, because I'm the benthic ecologist, I said, where did beluga get this selenium from? So we start to look at few organisms here. This part of the graph is exactly the same as I just showed you, but on a different shape. The beluga whale is just like rocket sky in this selenium. And one of the questions we have is it through the food web? We don't have the answer yet here. I can't tell you the answer. That's a project we started last year. So, but what is really important here, we have two major hypotheses that you can see. Why it's only in the skin of the beluga. I didn't have the time to show you the graph, but we spent some time with different Inuit community. And each time when they hunt for a beluga whale, when the animal is killed, they cut the tail, give the tail to the woman, go and have your party, and all the other group of hunters get together for the rest of the animal. So lucky enough, we can go in different groups. We collect some sample from the tail, from the skin, from the different part of the animal, and the tail is just like outrageously high in salinity. So, but each time the animal is flapping the sediment. So one of the question we have, could this, because these animals cannot secrete the salinity, it's come from some other little organisms such as bacteria or fungi. So is it the type of feeding habit? Is it through the food source? One of the questions, but why it's only in the skin? And though maybe during the molting season, where they are rubbing themselves in the sediment and in major algae, or like kelp. So that's one of the questions. Why I'm raising that is when you look here, the different village of Nunavut, that's 5 villages in Nunavut. We have also the animal living on the seafloor, and you can see that sea star are really almost at 1, which is exceptional in salinity. Same thing with the, again, so each time sea star are really high, but they are crawling in the sediment. We also have some data from the sediment presently, and that we're analyzing them, and it seems that even in the sediment, they are way higher. So maybe that's the sediment. That's a question. We have the same thing with mussel, but suddenly in urchin, which are just grazing, so it's still lower and the same level as the green sediment and the other one. So that's a question that we try to answer presently. But again, was to make an argument that even if the ice condition will change, we need to be careful. Inuit are not eating only maktak or beluga whale. Some other group are there, and they have also their value in food source. So just to recap, then to complete this conference, I hope I convinced you a little bit that the Arctic has a higher marine biodiversity than you should think straight at the beginning. The decrease is not in ice, it's not going to change the ecosystem, at least in a pelagic driven system, at least for the area we studied, the Nauplinia at that time. The Ventas got also some higher trophic level. It's not a black box of low trophic level, even if they are small now. And definitely the Inuit are using a larger range of marine resources. And what I showed the TTMU5, the one living closer to shore, are not going to be completely, I would say, replacing the beluga whale, but definitely can help with the salinity and all these different compounds which are still high in these animals. So thank you very much, Nakumi. Merci for this